would you like me to just go over what we're doing for today? We can do that. It's a good place to start. All right, so here we are. And we're in week 9. Week 8, we looked at something called mitosis. And we went through some of the different stages in mitosis. Oh, let me just make sure that I got everything going here so that I don't make a mistake. Make sure here. All right, we're sharing our screen. We're good to go. So what I wanted to just start with was here. So last week we looked at the cell cycle, and we looked at a particular stage, which was mitosis. And let's see if we can get this thing. Oh, it looks like it's writing backwards, so we'll have to fix that. So we looked at the cell cycle, and we focused on this phase here called the S phase, which was where DNA is replicated. And then we went on to this G2 phase, and we talked a little bit about that for the first couple days. And then we ended with the stages of mitosis. And I believe, let's go back to the class, you ended up with something that you had to draw. And I think this is it. Let's see. Yeah, and so we spent a little time reviewing this, and I kind of gave you some hints on sort of looking at the diagram and then trying to use this blank diagram and make inferences as to where the parts might fit. And what we did is we looked at how this is divided into 3 segments, and that there's one word that describes those 3 segments. And that here there were 4 things with one word describing that. And so I was trying to get you to look at that and look at this image. And there's 1, 2, 3. And remember, there's one word that describes the 3. So I wasn't giving you the answer at that time, but that now I can. It's interphase. And then we had mitosis and cytokinesis, which were these other stages that occur in the life cycle. And there was a little bit that you had to write. And then each of the stages, before we left, I had recommended going into this exact same thing here and looking at each of the different stages in a little bit more detail. Prophase, and then it goes to metaphase, which was number 2, and then anaphase. And in doing that, you would write a little bit of a description. And I'm pretty sure, I don't have it now, but I'm pretty sure when we left, I had done interphase and prophase and left you just with the last 2. And that's kind of where we ended last week. We had a little assessment at the end that kind of summarized all that. And so now that leaves us with week 9. And so it looks like you're going to be doing a week in review, sort of a recap. And so Mrs. Capetto came up with this, which I thought was kind of nice, was could you go through the meiosis chain of what actually happens in meiosis? So you're going to have to do a little bit of research here. The notes are really important. I'm not quite sure, and I'm going to, let's just check to make sure that those are actually in here. I do not see those. Why don't we make sure we do that right now? So I'll make sure at the end of this, I will add the notes in there so that you have access to the notes, because I think that's going to be something that you're going to need to be able to do this activity now that I look at it. So you'll do the recap, and then there's a Venn diagram, and in that Venn diagram, you're putting some of the words that both resemble what happens in mitosis and what happens in meiosis. In my opinion, what I would do is I would go back to last week and just take a look at mitosis at the end there. Look at the quiz that you did and see if you can go back into some of the vocab cards. You might see some of these same vocab words here. When I put the notes on, I think I have a nice little video clip that does a good comparison of mitosis and meiosis. That's from an amoeba sister, which I can see it looks like this comes from amoeba sisters. So meiosis, the great divide. Huh. So you know what? Let me see if I can find that video clip, and I will place that video clip in there for you so you have it as a resource. Why don't I do this? Why don't I just stop sharing for a moment? Okay, so what I did now is I added an additional instruction. I misnumbered, but that's okay. Do the video meiosis, the great divide, before completing the handout. I really think that is probably going to be the most beneficial to you. So if we watch just briefly. So they'll give you so this will do a good job because it's directly linked to that video. If you have a problem, or let me know, but it should connect right with it. So they're going in order right here. So what I would do is, as I'm watching the video, I would have that handout open up at the same time. And on that handout, it'll go through the same sequence, and then they kind of talk a little bit about it. So if you were working on that particular handout, when you get to that middle part, you'll be able to go through the sequence, and then the vocab words that they're using, you'll be able to use in here. Meiosis does do some very different things, and I put the notes in there for you, which are section 4. So it's intimidating because it looks like there is a lot to it, but I want to really just address the #1 issue with meiosis. I'm not going to get so wrapped up in the vocabulary right now, but where it is in red right here, this right here, is really important because what's really different about meiosis, and particularly metaphase, is this part right here. The chromosomes don't line up in a straight line like they do in mitosis. So when you look at mitosis, you will see that the chromosomes in metaphase line up in a straight line. See how they're all on top of one another? So that each one gets one of the chromosomes. One yellow, one blue, one green, one red. And this one will get one yellow, one blue, one green, one red. But in meiosis, that doesn't happen. You're going to end up with this cell having 2 reds and this one having a yellow. You see this little segment of red attached there. That's called crossing over. I'm not going to go into too much detail with that, but it is something that happens when chromosomes get a little too close to one another. But you'll see that is the big difference. And you'll notice that there is this thing says meiosis involves 2 divisions. There's a meiosis 1 and a meiosis 2. And so this process that you're looking at right here actually ends up happening twice. So this is meiosis 1, right? In mitosis, this is pretty much the end of it. But because of the way the chromosomes arrange themselves, there's another division because these are meant to form sex cells, the gametes, the sperm, and the eggs. And so you need to have only half of the genetic information because the other half of the genetic information comes from the sperm or the egg. And so it's not the same as mitosis because you're not trying to create a cell that has the exact same genetic information as the previous cell. What you're trying to do is create a cell that has half the genetic information so that when it meets up with another sex cell that has half the genetic information, they get together and then they create the correct amount of genetic information to have an organism. And then it develops. So mitosis and meiosis are very different in the sense that mitosis is to reproduce all of the cells in your body. Meiosis's purpose is to produce just the sex cells that have half the genetic information that is in each of your cells. So this process happens again. It's going to go through each one individually. A lot of it will look like mitosis, very, very similar. You have your spindle fibers. You've got your chromosomes. I actually put letters in here for you. And the reason I put the letters in here for you is this is where you start to begin to see how meiosis, this process, accounts for the Punnett squares that you may have worked on. Remember Gregor Mendel, the Punnett squares? So I put the letters in there to start getting kids familiar with capital A and lowercase a, and we can talk about dominant and recessive and start talking about traits and so forth. So remember I said that you need both chromosomes, one from mom and one from dad, and there was this idea of sister chromatids when they duplicated. So originally, the cell, before all of this happened, when the cell was a brand new cell, it had one chromosome A, capital A, and one chromosome lowercase a. Now, it looks like my pad here that I write on reset, so I have to use my mouse. And I think I can probably do, just briefly, maybe get this to work. So you've got this purple here. So what we'll do is we'll just grab, and we will grab, it's more like an eggplant color to me. Ooh, plum. That sounds good. And so you've got one chromosome that looks like that. That represents this plum color. And then there's this yellow color. So we've got that orangey yellow kind of color, gold, I guess. And that is over here. All right, and so again, they are the same length. So that's what we call sort of homologous in the sense that we're going to assume that they have the same genetic information, and they do. And this letter actually represents one of those genes. And so what you could do here, I don't know if I could, let's see. No, I can't, but I was going to try to put a letter A there. So let's just mark. We'll try the best we can. So if we put a mark here and we put a mark here, this one is a capital A. This is really tough to do with a, and this is a lowercase a. It's not too bad. And that's your trait. And that trait may be, say, I don't know, eye color. And that's the letter of the eye color that you get from dad, and that's the letter of the eye color you get from mom. And so you've got this capital and this lowercase, and then there's this idea of dominant and recessive. But what's really important is now we have this visual of these traits that are now represented by letters instead of just like imagining that they existed. So now when we say, okay, chromosomes duplicate and they're called sister chromatids, that's now because what you're saying is this chromosome duplicates, and on that spot would be another capital A. And on the other chromosome, it would duplicate. And on this spot right here would be another lowercase a. That's what sister chromatids are. They are duplicates of the chromosome that you got from dad and the chromosome you got from mom. So in this image, that's what you're looking at. You're looking at the homologous chromosome is the purple, and the gold would be homologous to each other. The sister chromatid is when we talk about the two purples because they're duplicate copies of one another, and these are duplicate copies. In mitosis, what would happen is the new cell would get one purple and one gold because that is what the original cell had. But in meiosis, our goal is not to produce a cell that works. Our goal is to produce a sex cell. So we don't want the sex cell to have a capital A and a lowercase a, or even two lowercase a's. We want the sex cell to have one of these, just one, either a capital A chromosome, which would be purple, or the gold chromosome, And then the other sex cell would bring in the other chromosome. So our purpose here is to end up with cells that have only one of these. So if you think about it, you should probably end up with 4 sex cells in this division process. Think about 4 sex cells, okay? This is just what crossing over is. You notice they have all the letters, and they're just showing that sometimes when these guys get really close to one another, they switch information. This just adds to genetic diversity. It's a little bit beyond the scope of what we need to talk about right now. Here's metaphase. Notice they're lined up next to one another. And what I did is I put mitosis next to the slide so that you could see the difference in the way they're arranged compared to this. Both these cells will exactly be the same. 1 gold, 1 blue, 1 green, and red. 1 gold, 1 blue, 1 green, 1 red. This is going to produce 2 purples, 2 golds, 2 greens, and 2 red, blues on each side, and that's not the same as the original cell. Cell separates through the process of anaphase, and then, of course, telophase, and then cytokinesis when they actually separate. But now we do this process again. Each of these, now remember we have capital A, and lowercase, lowercase. They separated. Each of these are actually going to go through that process again. Remember, here's our original cell. 1 red. We got 1 purple. Or 1 purple, 1 yellow, and 1 green. And now we've got cells that don't have all those colors. Instead, they have 4 chromosomes, because we have four chromosomes to start, but they don't have the right form chromosomes. They have a chromosome and its duplicate copy. Instead of having a different color, it has its copy of one of those, and it has another chromosome and its duplicate copy. So it has 4 chromosomes, but it doesn't have the 4 it needs, so it's not a viable cell. This is the original cell. So what happens here is each one of these cells are going to go through meiosis one more time. And they call that meiosis 2. And here is each cell going in its own path. And then what you'll see that does happen is here, you can see that in metaphase, in part 2, the chromosomes do line up in the same way that they do in mitosis, where instead of being next to one another, how they were in metaphase 1, they're now above one another so that they separate. But it's already too late. But remember, the purpose of this is to produce cells with half the genetic information because they're sex cells. And sure enough, look at how many cells we were able to produce. 1, 2, 3, and 4, which is what I had said before. 1, 2, 3, 4, half the genetic information. And then the process continues step by step, kind of goes into a little bit more detail. I get back to mitosis and meiosis comparison again at metaphase by showing you what meiosis 2 does, but how that compares. You can see that in meiosis 2, the lining up of chromosomes is similar, but different in meiosis 1. So if somebody asked you what part of meiosis is extremely different than mitosis, and you could say metaphase. Metaphase is the part of meiosis and mitosis that differs the most. And you end up with your 4. There is a word here, haploid, just means you have half the genetic information. So let's take a look at some of the vocab words that you have. Interphase, we can do. Process ends with identical cells. You could put that in. What process produces gametes? So gametes are sex cells. Produces 4 cells, each having 23 chromosomes. So for a human cell has 46 chromosomes, and I'm going to produce 4 cells. Which process, mitosis or meiosis, produced half of the number of chromosomes? In which process do chromosomes sometimes overlap and maybe exchange information? I just showed that in the notes from 11.4. So if it were me, I might go back to these and see if I can find a slide that shows chromosomes where it looks like they are crisscrossed. Oh, look at that. You can see how they're crossed and how a piece of this and a piece of the yellow just exchanged genetic information right here. Okay. Process is important for growth and repair. So which of the two processes, mitosis or meiosis, would be best suited for replacing cells in the event they were injured? And which one would be in the best for the process that produces cells that with another cell can make a new organism? So that's what they're getting at there. Again, the video will piggyback off of this assignment. I would watch those two simultaneously. Okay, so I don't think there should, I'm not sure if I should move on. You know what? Let me just, so that for tomorrow, just in case, let's make sure you know where you're going from this. Okay, so I do this mitosis, meiosis kind of thing, and I learn a little bit more about meiosis. I learn that metaphase is this one place where it's different. You're asking me to draw these illustrations and show the pathway. Where am I headed to tomorrow? Well, review textbook, complete the cell cycle and meiosis recap. And so what I'd like to do now is see if you could, if you're given a visual, could you compare and contrast? And so that is where we will be tomorrow. You can do this right in the document. Please make sure to make a copy and not to write into the original. And so Mrs. Capetta put this together, and she's trying to see if you can look at both and come up with things that happen in mitosis and things that happen in meiosis and things that happen in both. And so you can use the Venn diagram that you started today. And this is very similar in a Venn diagram because you have one side mitosis and you have one side meiosis, and then you have the overlap. And so essentially, you are making a more sophisticated Venn diagram with a little bit more information than you are in today's assignment. But that's essentially what you're doing. Two circles, and they're overlapping. And the both is where they overlap. And then on Wednesday, woohoo, you have a free day. So because I thought that meiosis is definitely an undertaking, it takes a little bit of patience and to think it through. I thought it would be appropriate to give students a little bit of extra time to work on the assignments for today and tomorrow, and also then on Wednesday. So I'm letting you know ahead of time that you know what I will do here. I will, you know what, why don't we keep them scheduled instead of opening them up, because that'll confuse everyone. So I'll keep it like this. Just know that since you're on today, you know that there's a flex day on Wednesday. And so there won't be any work due, but you'll have some extra time to complete the work from today and tomorrow. And we'll leave it at that. I'll have a live session tomorrow. I probably won't have much for Wednesday because there is no work due. I'll have mostly office hours. I think that's a perfect time for students to reach out. If you don't have any other questions, then what I'll do is I'll end this. And we'll stop recording.